A warm welcome to you. I'm really glad you're here today. Before we begin, I'd love for you to pop into the comments and let us know where you're listening from and what the local time is for you. It's always beautiful to see how global this little space has become. A big thank you to those who have subscribed and supported by liking videos. It truly helps this channel flourish. And if you'd like me to cover something in the future, your suggestions are always welcome. So now, let's begin. Chapter 1. Understanding Viruses, the Basics. Viruses are fascinating entities that occupy a unique place in the biological world. They are often described as the simplest form of life, yet they challenge our understanding of what it truly means to be alive. Unlike organisms that possess cellular structures, viruses are composed of only a few essential components, relying entirely on their host for replication and survival. They exist in a realm that blurs the lines between living and non-living. This paradox invites us to ponder, what exactly defines life? Can something so small and seemingly powerless wield such profound influence over living beings? At their core, viruses are made up of genetic material, which can be either DNA or RNA, encased in a protective shell known as a capsid. Some viruses are further enveloped in a lipid membrane, derived from the host cell's own membranes, which enhances their ability to infect new cells. This structure not only serves as a protective barrier, but also plays a critical role in the virus's ability to attach to and penetrate host cells. Imagine this protective shell as a tiny spaceship, designed to deliver its cargo of genetic instructions into the heart of a host cell. The capsid, composed of protein subunits called capsimers, is remarkable in its design. These proteins can assemble in various geometric arrangements, resulting in an array of shapes, from the icosahedral symmetry seen in many viruses to more complex helical forms. This structural diversity is not merely aesthetic. It reflects the virus's evolutionary adaptations to its environment and host. Each shape is a testament to nature's creativity and resilience, highlighting the intricate dance of survival that occurs at the microscopic level. The presence of an envelope distinguishes certain viruses from their naked counterparts. This lipid layer, often studded with proteins, facilitates the virus's entry into host cells, allowing it to masquerade as a familiar component of the cell's own structure. This clever tactic raises the question, how do viruses manage to deceive the immune system? The answer lies in their ability to mimic the very substances our bodies produce, effectively cloaking themselves from detection. When we delve into the classification of viruses, we encounter two primary types: DNA viruses and RNA viruses. DNA viruses, such as the smallpox virus, store their genetic information in the form of deoxyribonucleic acid. This stability allows for relatively slow mutations, leading to more consistent patterns of infection. In contrast, RNA viruses, like the influenza virus and the notorious human immunodeficiency virus, have a much higher mutation rate. This rapid evolution poses significant challenges for healthcare and vaccine development, as these viruses can swiftly adapt to evade immune responses. Imagine a game of hide-and-seek, where the seeker is perpetually outsmarted by a constantly changing target. The importance of studying viruses extends beyond mere curiosity. It has profound implications for public health and scientific advancement. Understanding the mechanics of viral infections enables researchers to develop effective treatments and vaccines. The global response to the COVID-19 pandemic has underscored the urgent need for knowledge in virology. Scientists, drawing on decades of research, mobilized their expertise to create vaccines at an unprecedented pace. This remarkable achievement reflects the cumulative effort of countless researchers, each contributing their unique insights and skills to combat a common enemy. Renowned virologist Dr. Anthony Fauci once stated, "The best way to prevent the next pandemic is to be prepared." This preparedness stems from an understanding of viral biology, transmission, and mutation. By exploring the myriad ways viruses interact with their hosts, we can begin to anticipate and mitigate future outbreaks. The study of viruses is not solely about the pathogens themselves. It is about the intricate relationships they forge with their environments and hosts, relationships that can lead to both illness and, paradoxically, advancements in medical science. As we immerse ourselves in the world of viruses, it becomes evident that they are not merely agents of disease. They are also tools of evolution. Viruses can transfer genetic material between different organisms, a process known as horizontal gene transfer. This mechanism can introduce new traits into populations, driving evolution in ways that are still being unraveled by scientists. For instance, the emergence of antibiotic resistance in bacteria has been linked to viral activity, highlighting the interconnectedness of all life forms. In our quest to understand these enigmatic entities, we might ask ourselves, what lessons can we learn from the viral world? One answer lies in the realization that viruses are a reminder of nature's complexity. They challenge us to reconsider our definitions of life and health, pushing the boundaries of scientific inquiry. Each discovery in virology opens doors to new questions, inviting us to explore further. Imagine for a moment gazing through a microscope, witnessing the interaction between a virus and its host cell. You might see the virus attaching to the cell's surface, its proteins binding like a key to a lock. As the viral genetic material enters the cell, it hijacks the cellular machinery, forcing the host to replicate the virus instead of its own components. This dramatic takeover is a microcosm of the struggle for survival that unfolds in every ecosystem. The study of viruses is a continuously evolving field, and as we advance our understanding, we uncover new strategies for combating viral diseases. Scientists are exploring innovative approaches, such as mRNA technology, which has shown great promise in vaccine development. By teaching our cells to recognize and respond to viral proteins, we empower our immune systems to defend against infections more effectively. This cutting-edge research exemplifies the potential of combining scientific knowledge with cutting-edge technology. As we reflect on the complexities of viruses, we find ourselves at the intersection of science and philosophy. What does it mean to coexist with entities that are both essential and harmful? In many ways, viruses challenge our perceptions of health, resilience, and adaptation. They remind us that life is a delicate balance, where the smallest players can have the largest impact. In the grand narrative of life on Earth, viruses play a vital role, shaping ecosystems, influencing evolution, and driving scientific discovery. They are not merely adversaries to be vanquished, but integral characters in the story of life itself. As we continue to unlock their mysteries, we tap into a wellspring of knowledge that can inform our future, guiding us in our efforts to promote health and well-being in a world that is constantly changing. With each revelation about viruses, we deepen our understanding of the biological world. We begin to see that these microscopic entities, often feared and misunderstood, are essential to the tapestry of life. They are both a challenge and a catalyst for innovation, urging us to explore the boundaries of what we know. As we embark on this journey through the fascinating realm of viruses, we remain ever curious, eager to uncover the secrets they hold. Within this intricate world of viruses, the dichotomy between DNA and RNA viruses becomes even more intriguing when we consider their life cycles. DNA viruses often have a relatively stable replication process, utilizing the host's cellular machinery to synthesize their own DNA and produce viral proteins. This process can lead to persistent infections, where the virus remains in a dormant state within the host, sometimes re-emerging later to cause illness. Imagine a long-hidden treasure lying in wait, only to be uncovered when conditions are just right. In contrast, RNA viruses typically operate with a different strategy. Their reliance on RNA, often characterized by a lack of proofreading mechanisms during replication, means that they can change rapidly. This high mutation rate allows them to adapt quickly to new environments and immune responses. One might envision them as chameleons, shifting colors to blend into their surroundings, evading detection and creating challenges for our immune systems and healthcare efforts alike. The HIV virus is particularly notorious for its ability to mutate and escape immune recognition, complicating treatment strategies. Consider the implications of these differences. The nature of a virus, whether it carries DNA or RNA, can dictate not only the severity of infections, but also the strategic approaches scientists must employ when developing vaccines and treatments. For instance, the stability of DNA viruses can lend itself to the development of more traditional vaccine strategies, while the dynamic nature of RNA viruses may call for more innovative approaches, such as those utilizing messenger RNA technology. The study of these viruses has led to remarkable breakthroughs in our understanding of disease and immunity. Dr. Robert Gallo, a pioneering figure in HIV research, emphasized the importance of understanding viral behavior when he noted, "A virus is only as good as its host." This insight sheds light on the intricate relationship between viruses and the organisms they infect, reminding us of the interdependence of life forms. As we contemplate the importance of studying viruses, it's essential to recognize their dual nature. While they can cause devastating diseases, they also serve as tools for scientific discovery. The techniques developed to study viruses have advanced fields such as genetics and molecular biology. By manipulating viral systems, researchers have been able to explore gene function and regulation, leading to discoveries that have profound implications for medicine and biotechnology. Imagine a laboratory where scientists harness the power of a harmless virus to deliver therapeutic genes to cells. This innovative approach, known as gene therapy, has the potential to treat genetic disorders and even some forms of cancer. The very same entities that can wreak havoc can also be transformed into vehicles of hope, showcasing the paradox of their existence. Moreover, viruses have played a significant role in the evolution of life itself. They have influenced genetic diversity across species, driving the adaptation of organisms through their interactions with host genomes. This phenomenon raises fascinating questions. How do viruses contribute to the resilience of life? Can they shape the future of species in ways we are only beginning to understand? As we ponder these questions, we may envision the viral landscape as a vast and ever-shifting frontier, where the interplay of infection, immunity, and evolution unfolds. This dynamic ecosystem not only challenges our understanding of health, but also inspires us to seek harmony within it. The balance between viruses and their hosts is a delicate one, and disruptions can lead to unforeseen consequences. In the grand scheme of public health, the study of viruses is essential. The emergence of novel viruses, as seen with recent global outbreaks, underscores the need for vigilance and preparedness. By studying the characteristics and behaviors of these pathogens, we equip ourselves to respond more effectively to potential threats. This proactive approach is a shared responsibility, one that involves scientists, healthcare professionals, and the public alike. Reflecting on the past, we can learn valuable lessons from the history of pandemics. Each outbreak has shaped our understanding of viral diseases and forged new paths in medical advancements. The swift development of vaccines during the recent pandemic is a testament to the resilience of science and the dedication of researchers worldwide. It exemplifies how knowledge and collaboration can converge to combat even the most formidable adversaries. As we journey deeper into the realm of viruses, it becomes clear that they are more than mere pathogens. They are complex entities that invite us to explore the boundaries of science and to question our definitions of life and health. Their study reveals not only the mechanisms of disease, but also the potential for healing and innovation. Envision yourself standing at the precipice of discovery, peering into a microscopic universe filled with both peril and promise. Each virus, each interaction, tells a story of survival, adaptation, and transformation. It is a narrative that unfolds in real time, echoing through the corridors of science and beckoning us to listen closely. In this exploration of viruses, we are reminded of the intricate web of life that binds us all. The very forces that challenge our existence can also lead to profound advancements in our understanding of biology and health. As we continue to unravel the mysteries surrounding these microscopic entities, we embrace the beauty of inquiry and the endless possibilities that lie ahead. Through this lens, we cultivate a sense of curiosity and wonder, eager to learn from the viral world while recognizing its role in our shared narrative. Each moment spent understanding these fascinating organisms brings us closer to a future where we harness their potential for the greater good, transforming fear into knowledge and adversity into opportunity. Chapter 2: How Viruses Find Host Cells. Viruses are remarkable in their simplicity, yet they possess an astonishing ability to find and invade host cells. This journey begins with a delicate interaction between the virus and the surface of the host. How does a virus, so diminutive and devoid of life as we know it, manage to locate its target among the vast array of cells? The answer lies in the mechanisms of viral recognition. Each virus has evolved unique strategies to identify potential host cells. One key aspect of this recognition is the interaction between viral proteins and specific molecules on the surface of host cells. Imagine a key fitting into a lock. This analogy beautifully captures the essence of how viruses attach to their hosts. The surface of a host cell is adorned with various receptors, each serving a distinct purpose in cellular communication and function. These receptors are not merely passive structures. They actively engage with external entities, including viruses. When a virus encounters a host cell, its surface proteins, often referred to as ligands, seek out compatible receptors. This specificity is akin to a dance of molecular recognition. For instance, the influenza virus employs hemagglutinin, a glycoprotein that binds to sialic acid residues on the surface of epithelial cells in the respiratory tract. It is this interaction that allows the virus to penetrate the cell membrane, facilitating entry and subsequent replication. This interaction prompts us to consider an intriguing question: what determines which hosts a virus can infect? The answer lies in a complex interplay of biological factors that influence host specificity. One significant factor is the genetic makeup of both the virus and the potential host. Viruses have evolved alongside their hosts, adapting to their unique cellular environments and immune responses. This coevolution shapes the landscape of viral infections, resulting in a remarkable specificity. Moreover, the presence and abundance of specific receptors on the host cell surface significantly influence this compatibility. For example, human immunodeficiency virus, or HIV, primarily targets T cells, a type of white blood cell crucial for immune function. The virus specifically recognizes the CD4 receptor along with coreceptors CCR5 or CXCR4, enabling it to gain entry into these cells. This specificity is pivotal, not only for the virus's survival, but also for the pathogenesis of the diseases it causes. It is fascinating to think about how viruses can evolve new strategies to overcome barriers posed by host defenses. Consider the case of the rabies virus, which has adapted to infect a wide range of mammals, including humans. Its ability to bind to nicotinic acetylcholine receptors has allowed it to exploit the nervous system, showcasing the remarkable adaptability of viruses in their quest for survival. Environmental factors play a role as well. Temperature, pH, and the presence of other cellular molecules can influence a virus's ability to recognize and bind to its host. For instance, some viruses are sensitive to temperature changes, which can affect their ability to infect specific tissues. This adaptability highlights the intricate relationship between viruses and their environments, suggesting that the search for hosts is not merely a biological process, but a dynamic one influenced by external conditions. Now, let us consider a few examples of viruses and their preferred hosts, each telling a story of adaptation and survival. The West Nile virus, for instance, primarily targets birds, particularly crows and jays, as its natural reservoirs. The virus circulates among these avian hosts, spreading to humans through mosquito vectors. This intricate cycle showcases how viruses can rely on specific hosts to thrive and propagate. In stark contrast, the poliovirus has a more selective approach. It primarily infects humans, thriving in the gastrointestinal tract before invading the central nervous system. The virus's reliance on human hosts reflects a fascinating evolutionary journey, ultimately leading to the development of vaccines that have drastically reduced its prevalence. As we explore these examples, it is essential to remember that viruses are not merely mindless entities. They are shaped by the environment and the delicate balance of life around them. They possess an uncanny ability to adapt, often displaying remarkable ingenuity in their search for new hosts. The relationship between viruses and their hosts is a reflection of nature's complexity. Each interaction is a reminder of the delicate balance that sustains life on Earth. Viruses can be agents of disease, but they also play significant roles in ecosystems, contributing to nutrient cycling and population control among various species. In the grand tapestry of life, viruses serve as both predators and prey, weaving themselves into the fabric of biological interactions. They remind us of the interconnectedness of all living beings, where even the smallest entities can have profound effects on the larger world. As we reflect on the journey of viruses in their pursuit of host cells, we cannot help but marvel at the intricate mechanisms that have evolved to facilitate this process. The delicate interplay of viral proteins and host receptors, influenced by genetic and environmental factors, speaks to the beauty of evolution. With each passing day, our understanding of these microscopic entities deepens, unveiling new insights into their behavior and impact. The scientific community continues to uncover the secrets that will allow us to better comprehend and combat viral infections. Imagine the future where we harness this knowledge to develop targeted therapies, vaccines, and strategies to mitigate the impact of viral diseases. The potential is vast, and the journey of discovery continues. Each revelation brings us closer to unraveling the mysteries of these enigmatic agents, enhancing our ability to coexist with them in a world that is both fragile and resilient. As we ponder the complexities of viral recognition and host specificity, we are reminded of the ongoing dance of life, where each entity plays its part in the grand narrative of existence. The exploration of viruses and their interactions with host cells is not merely an academic pursuit. It is a journey into the heart of life itself. Viruses exhibit an astounding level of specificity when it comes to the cells they infect. This specificity is largely determined by the presence of unique receptors on the host cell surfaces. These receptors serve as the gateways for viral entry, and their distribution varies significantly across different types of cells and organisms. Picture a vast landscape where each cell type is a unique dwelling, equipped with its own set of doors, some welcoming and others firmly shut. The intricacies of these receptors can be quite remarkable. For instance, the receptors on the surface of a human liver cell differ fundamentally from those on a skin cell. This diversity allows some viruses to be highly selective, honing in on particular tissues and organs. The hepatitis B virus, for example, has a particular affinity for liver cells, where it can replicate and cause disease. The interaction between the virus and its host receptor is not random. It is a finely tuned process shaped over millennia of evolution. Have you ever considered how viruses might choose their hosts? This process is influenced by a combination of genetic compatibility and environmental factors. The molecular structure of both the viral proteins and the host receptors plays a crucial role in this interaction. If we think of the virus as a key, then the host cell's receptor is the lock that it seeks to turn. When the shapes and chemical properties align, the door to infection swings open. Another fascinating aspect of this interaction is how viruses can adapt to changes in their environment or host population. Consider the human influenza virus, which undergoes a process known as antigenic drift. This phenomenon allows the virus to make small changes to its surface proteins, helping it evade the immune system and persist in the population. Imagine a chameleon changing its colors to blend into its surroundings. This adaptation enables the virus to continue its existence while maintaining its ability to infect. The concept of coevolution also comes into play. As viruses evolve to better infect their hosts, those hosts simultaneously develop defenses to combat these invaders. This ongoing struggle between virus and host can lead to a fascinating arms race, where both parties are in a constant state of adaptation and response. The interplay is not merely a battle for survival. It's a complex relationship that can influence the evolution of entire species. Exploring the dynamics of host specificity further, we encounter the concept of zoonotic viruses, those that jump from animals to humans. The emergence of viruses such as Ebola or Zika has sparked interest in how these pathogens can cross species barriers. Often, the initial hosts are wild animals, and the transmission to humans can occur through direct contact or via vectors like mosquitoes or bats. This highlights the importance of understanding ecological contexts and how human encroachment into wildlife habitats can facilitate these viral transmissions. The story of the Nipah virus serves as an illustrative example of how zoonotic viruses can emerge. Initially identified in Malaysia, it is believed that fruit bats were the natural hosts, with pigs acting as intermediate hosts before infecting humans. The virus showcases the potential consequences of disruptions in ecosystem balance, as increased interactions between wildlife and agricultural practices can lead to the emergence of new diseases. As we reflect on these various examples, it becomes clear that a multitude of factors influence which viruses can infect which hosts. In addition to genetic and ecological factors, the immune status of a potential host plays a significant role. Some individuals may carry genetic variations that confer resistance to specific viral infections, while others may be more susceptible due to underlying health conditions or weakened immune responses. The interconnectedness of life on Earth further complicates our understanding of viral host specificity. Viruses can act as agents of change, influencing population dynamics and driving evolution in their hosts. Consider the bacteriophage, a virus that infects bacteria. These tiny entities are prolific in nature, controlling bacterial populations and thereby shaping ecosystems. In this light, viruses can be viewed as not only pathogens, but also as essential components of biological communities. The elegance of viral recognition and host specificity serves as a reminder of the complex web of life that surrounds us. Each interaction between a virus and its host cell is a testament to the ongoing processes of evolution and adaptation that define our world. As we continue to explore these microscopic entities, we uncover the layers of complexity that govern their existence and our own. What lies ahead in our understanding of these relationships? The future promises exciting developments as researchers delve deeper into the molecular mechanisms of viral host interactions. By studying these processes, we gain insights that could lead to innovative therapeutic strategies and vaccines, ultimately enhancing our ability to manage viral infections. Imagine a world where we can predict and prevent viral outbreaks through a comprehensive understanding of host specificity. This vision is not merely a dream. It is a possibility that could reshape our approach to global health. The knowledge we gather today will pave the way for a more resilient future, where humanity can coexist with viruses in a balanced and harmonious manner. As we contemplate the myriad of factors influencing viral recognition and host interactions, we stand at the threshold of discovery. With each new piece of knowledge, we move closer to unlocking the secrets of these enigmatic agents, revealing the profound impact they have on life as we know it. Chapter 3: The Attachment Phase of Infection. The journey of a virus toward infection is both intricate and deliberate, marked by a crucial phase that sets the stage for its next steps, the attachment phase. This phase is where the virus makes its initial contact with a host cell, a moment that holds profound significance for both the invader and the invaded. When a virus approaches a host cell, it embarks on a meticulous process of attachment, a choreography that requires precision and adaptability. The first step involves the virus utilizing its surface proteins, which play a pivotal role in binding to specific receptors located on the host cell's membrane. These viral proteins function as the keys that fit into the locks of cellular receptors, enabling a secure connection. Imagine this interaction as a handshake, where both parties recognize each other's presence and intentions. The importance of viral proteins in binding cannot be overstated. These proteins are not mere structural components. They are sophisticated tools honed by evolution to ensure successful infection. Each type of virus has evolved distinct proteins tailored to target specific host receptors. For example, the human immunodeficiency virus, or HIV, employs a surface protein called GP120 that binds to the CD4 receptor on human T cells. This binding is not just a casual touch. It triggers a cascade of events that facilitate the virus's entry into the cell. As the late virologist Max Delbruck once said, the virus is a very simple organism. It has a very simple life. Yet, within that simplicity lies a complex mechanism that has evolved over eons. Variations in attachment strategies among different viruses add further layers of intrigue to this process. Some viruses, like the rhinovirus responsible for the common cold, utilize a different approach. They attach to host cells through interactions with intercellular adhesion molecules. This diversity in strategies showcases the adaptability of viruses as they exploit the unique characteristics of their environments. Different viruses have carved out niches, each developing methods that maximize their chances of successful attachment and subsequent infection. The consequences of attachment extend far beyond the initial binding. Once a virus has successfully attached to a host cell, it sets off a series of biological responses that can have profound implications for the cell's fate. The binding of the virus often initiates a process called receptor-mediated endocytosis, where the host cell engulfs the virus in a bubble-like vesicle. This is akin to a cell pulling in a small package, inviting the virus deeper into its interior. However, the aftermath of this event can be dire for the host cell. The presence of the virus can trigger an immune response, leading to inflammation and cellular stress. The cell, recognizing that it has been compromised, may begin to activate its defenses, attempting to eliminate the intruder. Imagine a fortress under siege, where the guards scramble to respond to an unexpected attack. Yet, in the case of viral infections, the line between defense and damage can blur. The attachment phase is therefore a double-edged sword. While it is the necessary precursor to infection, it also marks the beginning of a struggle between the virus and the host's immune system. In some cases, the viral attachment can lead to cellular changes that enable the virus to hijack the cell's machinery, transforming it into a factory for viral replication. This transformation can be likened to an artist commandeering a canvas, turning a once innocent space into a masterpiece of viral propagation. As we consider the complexities of the attachment phase, we might ask ourselves, what happens to the host cell after the virus has taken hold? The answer reveals a landscape of cellular disruption. The host cell may experience changes in its normal functions as the virus redirects resources to support its own replication. This diversion can lead to cellular death, a sacrifice made in the face of an overwhelming viral onslaught. Furthermore, the attachment phase can also influence the host's immune response. The nature of the viral proteins can determine how effectively the immune system recognizes and responds to the invasion. Viruses like the influenza virus are notorious for their ability to change their surface proteins through a process called antigenic drift. This continuous evolution allows them to evade detection, making it even more challenging for the immune system to mount an effective defense. The variability in viral attachment strategies and their consequences underscore the delicate balance of life and infection. The moment a virus attaches itself to a host cell is not just a mere interaction. It is a turning point that can dictate the course of an infection. Each attachment is a story, unique in its details and outcomes. In the grand scheme of biology, the attachment phase of infection serves as a reminder of the intricate relationships between organisms, the struggle for survival, and the resilience of life in its many forms. As we ponder these dynamics, we can appreciate the remarkable adaptability of both viruses and their hosts, each engaged in a timeless dance of survival. The science of viruses is a journey that offers endless fascination, revealing the wonders and challenges of life at the microscopic level. Through the lens of this attachment phase, we glimpse not only the mechanisms of infection, but also the broader implications for health and disease. The ability of viruses to attach and invade is a powerful testament to their evolution, resilience, and impact on the living world. The next stage in this intricate saga is one of replication and spread, where the consequences of attachment unfold with each passing moment. As the viral particles attach themselves to the host cell, they begin to exploit the very mechanisms that sustain the life of the cell. This phase of attachment is not just a solitary event. It unfolds with a series of interactions that are as intricate as they are vital. The binding of viral proteins to host receptors can initiate several pathways within the host cell, leading to a cascade of cellular responses. When the virus successfully latches onto its target, the host cell's signal transduction pathways become activated. These pathways are like intricate wires within a complex system, transmitting messages that can alter cellular behavior. For some viruses, the attachment phase can trigger changes in the host cell's membrane potential, making it more permeable and conducive to viral entry. Imagine a dam bursting open, allowing for an influx that was previously restrained. The mechanisms by which viruses can alter host cell functions vary widely. Some viruses induce the production of specific proteins that facilitate their own entry, while others may manipulate the host's cytoskeletal dynamics, allowing the virus to be engulfed more efficiently. This manipulation is a testament to the virus's cunning adaptability. For example, the vesicular stomatitis virus employs a fusion protein that enables its membrane to merge with the host's, creating a passageway into the cell's interior. Moreover, the consequences of attachment extend beyond mere entry. The act of binding can also alter the host cell's gene expression. When viruses attach to their receptors, they can provoke the activation of transcription factors within the cell. These factors can lead to the expression of genes involved in inflammation or, conversely, can suppress the expression of antiviral responses. This duality underscores the strategic prowess of viruses, as they navigate the delicate balance of evading the immune response while simultaneously commandeering the host's resources for their replication. This interplay between the virus and the host cell raises an important question. How do different viruses fine-tune their strategies to ensure successful attachment and entry? The answer lies in the evolutionary pressures that have shaped each virus's interaction with its host. Over countless generations, viruses have adapted to their environments, developing mechanisms that optimize their chances of infection. Consider the varicella-zoster virus, the causative agent of chickenpox. This virus utilizes a glycoprotein called GB to attach to host cells. Remarkably, this attachment not only facilitates entry, but also allows the virus to evade the host's immune surveillance. The ability to remain latent in host cells after initial infection speaks to the virus's evolutionary success. The delicate balance between infection and immunity is a dance influenced by time, adaptation, and survival. The attachment phase also opens up discussions about the potential for therapeutic interventions. Understanding the molecular details of how viruses attach to cells can provide insights into developing antiviral drugs. Imagine if we could design inhibitors that block the very proteins responsible for this vital interaction, much like placing a barrier that prevents a thief from entering a home. Such strategies could significantly diminish the ability of viruses to infect host cells, providing a layer of protection for vulnerable populations. The repercussions of viral attachment are not restricted to the immediate fate of the host cell alone. Once a virus has successfully attached and begun its invasion, it can trigger a broader immune response within the organism. The innate immune system, acting as the body's first line of defense, recognizes the presence of foreign invaders. Cells such as macrophages and dendritic cells play pivotal roles in this response, sensing the viral proteins on the surface of infected cells. This detection can lead to the release of signaling molecules called cytokines, which orchestrate an inflammatory response. The cytokine storm, while essential for combating infection, can also lead to tissue damage and contribute to the severity of the illness. In contemplating the attachment phase from the perspective of the host, we must also consider the psychological ramifications of viral infections. The anxiety and fear that accompany the onset of illness often stem from an awareness of the unseen battles taking place within our bodies. As one contemplates the minute viral particles attaching to cells, it can evoke a sense of vulnerability. Yet, within this vulnerability lies the potential for resilience. The human immune system is equipped with a remarkable capacity to learn and adapt, forming a memory of past infections that can inform future responses. As we delve deeper into the consequences of attachment, we can connect these biological events to broader public health implications. The dynamics of viral infections, particularly during outbreaks, highlight the importance of understanding how viruses attach to host cells. The ability to predict and respond to viral threats hinges on our comprehension of these fundamental processes. The intricate symphony of attachment, immune response, and cellular alteration illustrates not only the power of viruses, but also the resilience of life. Each interaction is a reminder that our existence is intertwined with these microscopic entities. In this dance of survival, the attachment phase serves as a critical juncture, one that shapes the trajectory of infection and disease. As we ponder the myriad ways in which viruses attach to their hosts, we also recognize the elegance of evolution at play. The complexity of these interactions invites us to celebrate the diverse strategies employed by viruses while acknowledging the remarkable adaptability of the host. The story of attachment is not merely one of invasion. It is a narrative woven into the fabric of life itself, where each attachment holds the potential to change the course of both viral and cellular destinies. In this delicate interplay, we find a reflection of the broader themes of existence: resilience, adaptation, and the relentless pursuit of survival. As we continue to explore the intricate world of viruses, we remain mindful of the profound relationships that govern life, urging us to delve further into the mysteries that lie ahead. Chapter 4: Penetration: How Viruses Enter Cells. As we journey further into the world of viruses, we arrive at a pivotal chapter: the moment they penetrate the very cells they seek to inhabit. This crucial act of entry is not merely a straightforward passage. It is a complex ballet of biological interactions involving various methods and mechanisms that have evolved over millennia. Two primary methods of viral penetration stand out: fusion and endocytosis. Imagine fusion as a gentle embrace, where the viral envelope merges seamlessly with the host cell membrane. This process occurs primarily in enveloped viruses, which carry a lipid bilayer derived from their host. When the virus approaches a cell, specific proteins on the viral envelope facilitate this merging, allowing the viral contents to spill into the cytoplasm. It is reminiscent of a whisper, where the delicate exchange of materials signifies an intimate connection between the virus and its host. In contrast, endocytosis presents a different but equally fascinating approach. Picture a cell extending its arms, enveloping the virus within its membrane like a protective cocoon. This process is especially prevalent among non-enveloped viruses, which lack the lipid layer that enables fusion. Upon binding to the cell surface, the virus triggers a cascade of events that leads to the invagination of the membrane, ultimately encapsulating the virus in an endosomal vesicle. This vesicle then travels deeper into the cell, setting the stage for the viral genetic material to be released and commandeer the host's machinery. The mechanisms of membrane disruption employed by viruses are as varied as the viruses themselves. Some viruses utilize the physical properties of their proteins to poke holes in the host membrane, while others may release enzymes that degrade membrane components. For instance, the influenza virus, once inside the endosome, employs an ingenious trick. It exploits the acidic environment of the endosome to activate fusion proteins, allowing the viral envelope to merge with the endosomal membrane. This clever adaptation ensures the virus can escape into the cytoplasm, illustrating the remarkable ingenuity of these microscopic entities. The role of cellular machinery in facilitating viral entry cannot be understated. Host cells are not passive spectators in this process. Rather, they provide an array of tools that viruses cleverly exploit to their advantage. Once a virus has successfully entered the cell, it manipulates cellular pathways to its benefit. For example, some viruses hijack the host's cytoskeletal system, using it as a transport network to move within the cell. Imagine this as a bustling city, where the virus commandeers the existing infrastructure to navigate through the cellular landscape, seeking out the nucleus where it can replicate and thrive. This interplay between viral strategies and host cellular machinery varies widely across different virus families. Each virus adapts its method of entry to suit its unique biological makeup and the specific characteristics of the host cell it targets. For example, the human immunodeficiency virus employs a sophisticated mechanism that involves not only the binding of its surface proteins to CD4 and coreceptors, but also the subsequent internalization of the entire complex into the host cell. This nuanced approach highlights the evolutionary arms race between viruses and hosts, driving each to develop ever more sophisticated methods of interaction. Consider the adenovirus, a non-enveloped virus that employs a different strategy altogether. After binding to its specific receptors, it utilizes a fiber protein that facilitates the virus's entry through endocytosis. Once inside, adenovirus can escape from the endosome and directly access the cytoplasm, showcasing yet another method of membrane disruption. The diversity in strategies is a testament to the adaptability of viruses, each uniquely equipped to penetrate cellular defenses. As we explore these varied methods of penetration, we begin to appreciate the intricate relationship between viruses and their hosts. It raises profound questions about the nature of life itself. How do such simple entities possess such complexity in their means of survival? What drives these microscopic invaders to evolve such elaborate mechanisms for entry? Each question invites us to reflect on the delicate balance of life and the unseen battles that occur at a cellular level. In our quest to understand these processes, we must also acknowledge the contributions of remarkable scientists who have dedicated their lives to unraveling these mysteries. Figures like Albert Calmette, who, in the early 20th century, dedicated his research to understanding viral infections, laid the groundwork for our current knowledge. His explorations into the dynamics of viruses and their interactions with host cells opened the door to a broader understanding of infectious diseases and their implications for human health. The study of viral penetration is not merely an academic exercise. It has real-world implications. Understanding how viruses enter cells can inform vaccine development and therapeutic strategies. For instance, by deciphering the precise mechanisms of entry, researchers can design targeted interventions that block these pathways, effectively preventing infection before it can take hold. Imagine a world where we can anticipate a virus's moves, thwarting its attempts at invasion with precision. This vision is closer to reality than ever before, thanks to the tireless work of scientists worldwide. Their insights illuminate not only the pathways of viral entry, but also the broader implications for our understanding of health and disease. As we delve into the mechanisms of viral penetration, we uncover a world rich with detail and complexity. Each interaction, each method of entry, tells a story of evolution, adaptation, and survival. By understanding these processes, we draw closer to a deeper comprehension of life itself, how even the smallest entities can wield power over the vast realm of living organisms. In this ongoing narrative, the virus emerges not simply as an invader, but as a participant in the grand cycle of life, influencing and being influenced by the very cells it seeks to penetrate. The intricate dance of viral penetration reminds us of the interconnectedness of all living things, even amidst the struggle for survival. As we continue to explore this captivating world, we remain ever curious about the stories that unfold within the microscopic realms that govern our existence. The methods of viral penetration reveal a fascinating interplay between the simplicity of viruses and the complexity of cellular machinery. As we delve into the mechanisms of membrane disruption, we encounter a multitude of strategies that viruses employ to gain access to their host cells. Some viruses have evolved to utilize physical properties inherent in their structure, while others deploy sophisticated biochemical tactics. Consider the remarkable ways in which the herpes simplex virus operates. This virus, known for causing cold sores, employs a unique mechanism for entry. It exploits the host cell's machinery by first binding to specific receptors, leading to the fusion of the viral envelope with the cell membrane. The viral glycoproteins serve as guides, orchestrating the fusion process with precision. Imagine these proteins as skilled conductors, directing a symphony of molecular interactions that culminate in the opening of a portal for the viral genome to enter the host's cytoplasm. In contrast, some viruses, such as the poliovirus, take a different route altogether. Once it attaches to its receptor, poliovirus undergoes a conformational change that allows it to inject its RNA directly into the host cell. This highly efficient method bypasses the need for endocytosis, demonstrating that viral strategies can be as diverse as the viruses themselves. What drives these variations in penetration methods? Evolution has finely tuned these viruses to maximize their chances of survival within specific environments and host organisms. The role of cellular machinery in facilitating entry is a testament to the intricate relationship between viruses and their hosts. Viruses are not merely invaders, they are adept manipulators of cellular processes. After gaining entry, they often hijack the host's replication and protein synthesis systems to produce copies of themselves. This phenomenon raises an intriguing question. How do viruses manage to exploit these complex cellular pathways without being detected by the host's immune defenses? The answer lies in the virus's ability to mimic cellular processes. For example, the hepatitis C virus camouflages itself within lipid droplets, utilizing the host's own lipid metabolism as a shield against immune detection. This clever adaptation allows the virus to persist within the body, evading the immune response while it replicates. As we explore the variability of penetration strategies across virus families, it becomes evident that each virus has tailored its entry process to fit its unique ecological niche. The influenza virus, with its segmented RNA genome, employs a dynamic approach, relying on both receptor-mediated endocytosis and fusion. This flexibility provides the influenza virus with a distinct advantage, enabling it to adapt rapidly to different hosts and environmental conditions. Similarly, the Ebola virus exhibits an intricate strategy that highlights its virulence. Upon binding to host cell receptors, it triggers endocytosis, leading to the formation of vesicles that transport the virus deep into the cell. Once inside, The virus employs a mechanism to escape the endosome, allowing it to release its genetic material into the cytoplasm. This swift penetration is critical, especially given the virus's rapid replication cycle and the severe consequences of infection. The variation in penetration strategies invites us to consider the evolutionary pressures that shape these processes. Why do certain viruses adopt such elaborate entry mechanisms while others appear more straightforward? The answer may lie in the host's defenses. Viruses that infect more complex organisms must navigate a host of sophisticated immune responses. In this context, the complexity of their entry strategies serves as a countermeasure against the host's attempts to thwart infection. This delicate dance between viruses and host cells evokes reflection on the broader implications of viral behavior. Each successful entry not only signifies a potential new infection, but also underscores the ongoing evolutionary arms race between pathogens and their hosts. The development of antiviral therapies and vaccines hinges on our understanding of these processes. Imagine a future where we can anticipate and preempt viral invasions, crafting defenses that are as dynamic as the viruses themselves. The contributions of scientists in this field have been monumental. Researchers like Michael Rossman, who has elucidated the structures of various viruses, have provided invaluable insights into how viruses penetrate cells. His work on the structure of the poliovirus has revealed critical details about the mechanisms behind its entry. By understanding the structure, we can begin to understand function, he noted, highlighting the importance of structural biology in unraveling the mysteries of viral penetration. As we immerse ourselves in the nuances of viral entry, we uncover stories of survival, adaptation, and the relentless pursuit of existence. Each virus tells a tale of evolution, shaped by the challenges it faces and the strategies it devises. In this realm, viruses are not merely agents of disease, they are entities that embody the fundamental principles of life itself: adaptation, interaction, and persistence. The journey of a virus as it penetrates a host cell is a microcosm of the larger themes of life. It reminds us that even within the smallest forms of life, there exists a complexity that invites wonder and curiosity. As we continue to explore the mechanisms of viral penetration, we remain in awe of the intricate interplay between these microscopic entities and the cells they seek to inhabit. The stories they weave are not just about infection. They are reflections of the interconnectedness of all living organisms, a reminder that life, in all its forms, is a continuous and dynamic process. Chapter 5: Unpacking Viral Genetic Material Inside Hosts. As the viral particles settle within the host cell, a new phase of their journey begins: the release of their genetic material. This pivotal moment marks the transition from mere presence to active engagement. Imagine the viral capsid, which has been a steadfast guardian, now unlocking its secrets, allowing the genetic material to flow into the cytoplasm. This release is akin to a treasure chest opening, revealing the blueprints of life that the virus carries within. The mechanisms of uncoding, as this process is termed, vary among different viruses yet share a common objective: to liberate the viral genome for replication. Uncoding can occur through various means, such as the action of host cell enzymes or the environment within the cell that triggers the capsid to disassemble. Consider the enveloped viruses, which often employ the very lipids of the host cell to facilitate this uncoding. It's as if they are borrowing a part of their host's identity to advance their own replication. For instance, once the influenza virus enters a host cell, the acidic environment within the endosome triggers conformational changes in the viral proteins. This process leads to the fusion of the viral envelope with the endosomal membrane, allowing the viral RNA to be released into the cytoplasm. The elegance of this mechanism is a testament to the virus's evolutionary adaptation to exploit its environment for survival. The significance of uncoding cannot be overstated. It is during this act that the viral genome is made accessible, enabling the virus to hijack the host's cellular machinery. Without uncoding, the viral genetic material would remain dormant, a mere whisper in the cacophony of cellular activity. This phase is not just a mechanical process. It is a calculated move in the viral strategy to ensure its survival and proliferation within the host. Once released, the viral genome must be replicated to produce new viral particles. This replication is crucial for the continued spread of the infection. Different viruses employ varied strategies to achieve this goal, reflecting the diversity and adaptability of these entities. For example, DNA viruses, like the herpes simplex virus, utilize the host's DNA polymerase to replicate their genetic material, seamlessly integrating their needs with the host's cellular processes. In contrast, RNA viruses, such as the Ebola virus, often replicate their genomes in the cytoplasm using viral enzymes, a more self-reliant approach that showcases their unique adaptations. Imagine the viral replication process as a factory assembly line, where each component is meticulously crafted to ensure the production of new viral entities. The viral genome serves as both a template and a guide, directing the synthesis of viral proteins and new genetic material. This process is not merely about multiplication. It is about creating a new generation of viruses, each equipped to infect additional host cells. Moreover, the replication strategies employed by different viruses reveal a fascinating aspect of their biology. Consider the retroviruses, such as HIV, which utilize reverse transcription to convert their RNA genome into DNA. This newly formed DNA can then integrate into the host's genome, a stealthy method of ensuring that the virus persists even when host cells divide. The complexity of this strategy raises profound questions about the interplay between viruses and their hosts. How do these viral tactics influence the evolution of host defenses? As we explore the differences in replication strategies, the diversity of viral approaches becomes apparent. Some viruses, like the poliovirus, replicate swiftly, producing multiple progeny within a matter of hours. Others, such as the human papillomavirus, may linger in a latent state, evading detection and immune responses for extended periods. This variation in replication rates and strategies not only highlights the adaptability of viruses, but also underscores the ongoing battle between viral invaders and the immune defenses of their hosts. The significance of understanding these replication strategies extends beyond the realm of virology. It offers insights into potential therapeutic approaches. By targeting specific stages of viral replication, scientists can develop drugs that inhibit viral spread, providing a crucial line of defense against infections. As we immerse ourselves in this intricate world, consider the role of the scientists who have dedicated their lives to unraveling these mysteries. Individuals like Albert Sabin, who developed the oral polio vaccine, exemplify the profound impact of understanding viral biology. Their work not only protects countless lives, but also illuminates the path forward in combating viral diseases. The interplay of viral genetic material and host cellular processes paints a picture of life at the molecular level, where every interaction holds significance. Each replication cycle opens new avenues for exploration, inviting us to ponder the boundaries of life and the resilience of these microscopic entities. In this ongoing narrative of infection, the release of viral genetic material and subsequent replication serves as a reminder of the delicate balance within ecosystems, where viruses play both destructive and constructive roles. They are not merely harbingers of disease, they are agents of change, influencing the evolutionary trajectory of their hosts. As we reflect on this intricate dance of life, one may wonder, what lessons do these viruses hold for us? The answers lie in the depths of their genetic diversity and adaptability, revealing a world that is both fascinating and complex. With each cycle of uncoding and replication, viruses showcase their remarkable ability to thrive in a host environment. Their strategies, honed over countless generations, remind us of the resilience inherent in all forms of life. This resilience serves as a testament to the intricate connections that bind every organism, no matter how small or seemingly insignificant. In the grand tapestry of life, viruses occupy a unique niche, inviting us to explore the boundaries of our understanding. As we continue to learn from their strategies and impacts, we embrace the challenge of coexisting with these remarkable entities that, despite their simplicity, have the power to reshape our world. As the viral genetic material finds its way into the host cell, the next crucial step unfolds: the replication of this genetic blueprint. This process is not merely a mechanical act. It demands precision, timing, and an intricate understanding of the host's cellular machinery. The viral genome, once liberated, becomes the master template from which new viral particles will emerge, each crafted with the intention of perpetuating the infection. At this juncture, one might wonder, how does a virus ensure that its genetic material is replicated accurately? The answer lies in the sophisticated interplay between the viral genome and host enzymes. For DNA viruses, the replication process often mimics that of the host's own DNA replication. The viral DNA, upon entering the nucleus, commandeers the host's DNA polymerase. This enzyme, typically responsible for copying the host's genetic information, is repurposed to create copies of the viral genome. Imagine a skilled locksmith using a key not just to open a door, but to create duplicates of that key. This remarkable mimicry allows the virus to harness the host's cellular machinery while minimizing its own complexity. Conversely, RNA viruses face a different challenge. Lacking the sophisticated machinery found in DNA viruses, they often utilize their own RNA-dependent RNA polymerase to replicate their genomes directly in the cytoplasm. This enzyme, which the virus encodes, facilitates the synthesis of complementary RNA strands. Picture a master artisan crafting intricate designs, each strand representing a new generation of viral RNA, ready to be packaged into new viral particles. The rapid replication of RNA viruses underscores their evolutionary advantage, enabling them to produce vast quantities of progeny in a short span of time. As we consider the replication strategies employed by viruses, the diversity among them is striking. Some viruses, like the rhinovirus, replicate at an astonishing rate, often overwhelming the host's defenses before they can mount a response. Others, such as the hepatitis B virus, may establish a chronic infection, integrating their DNA into the host's genome and eluding detection for years. This ability to adapt and thrive in various environments speaks volumes about the evolutionary pressures that shape viral behavior. Throughout this intricate process, the virus must also contend with the host's immune responses. The immune system stands as a formidable adversary, equipped with various strategies to detect and eliminate viral invaders. This ongoing battle creates a dynamic environment where viruses must continually evolve to evade detection. The strategies employed by viruses to escape immune surveillance are as diverse as the viruses themselves. Some viruses, like the cytomegalovirus, develop intricate mechanisms to inhibit the presentation of viral antigens on the surface of infected cells, effectively hiding from the immune system. Others, such as influenza, undergo rapid mutations, resulting in new viral strains that can evade previously established immunity. In this context, consider the work of scientists like Paul Offit, who has dedicated his career to understanding and combating viral infections. His insights into the mechanisms of viral replication and immune evasion have paved the way for innovative vaccine strategies that can bolster our defenses against these cunning pathogens. The significance of understanding viral replication extends far beyond the realm of virology. It holds the potential for therapeutic breakthroughs that can mitigate the impact of viral infections on public health. By unraveling the intricacies of viral life cycles, researchers can identify critical points of intervention, enabling the development of antiviral drugs that disrupt specific stages of replication. As the narrative of viral infection unfolds, it becomes evident that the interplay between viral replication and host responses is a delicate balance, reminiscent of a complex ecosystem. Viruses, in their relentless pursuit of survival, act as both agents of disease and catalysts for change within their environments. They challenge our understanding of life and force us to reconsider our relationship with these microscopic entities. With every cycle of replication, we witness the resilience and ingenuity of viruses. Their ability to adapt and thrive amidst the challenges posed by host defenses exemplifies a remarkable aspect of life: the relentless drive to persist. As we reflect on this intricate web of interactions, we are reminded of the profound interconnectedness of all living organisms, where even the smallest entities can have far-reaching effects. In contemplating the various strategies employed by viruses, one might ask, what can we learn from these resilient organisms? Each viral adaptation offers a lesson in survival, illustrating the importance of flexibility and innovation in the face of adversity. As we delve deeper into the world of viral genetics, we uncover insights that can inform not only our approach to combating infectious diseases, but also our broader understanding of life's complexities. In this ongoing exploration, the replication of viral genetic material inside hosts serves as a reminder of the intricate dance of life at the molecular level. Each interaction, each replication cycle, unfolds like a story, revealing the rich tapestry of existence that binds us all. As we continue to unravel these biological narratives, we embrace the challenge of coexisting with these remarkable entities, recognizing their role in shaping the world around us. Chapter 6: Viral Replication, Taking Over the Host Machinery. The journey of a virus does not end with the uncoding of its genetic material. Rather, it marks the beginning of a new chapter in the viral life cycle: replication. Once the viral genome is released into the host cell's cytoplasm, it embarks on a mission to exploit the cellular machinery, orchestrating a series of intricate steps that ensure its survival and proliferation. The replication cycle of a virus can be divided into several distinct phases: attachment, penetration, uncoding, replication, assembly, and release. Each of these phases plays a crucial role in the virus's ability to hijack the host's cellular processes. Imagine a skilled conductor leading an orchestra, where each musician represents various components of the host cell, responding to the viral instructions with precision and timing. The virus, though lacking the ability to replicate independently, has evolved to manipulate the host's cellular systems, turning them into factories for viral production. Initially, the viral genome must be transcribed and translated into proteins. This step is pivotal, as the host cell's ribosomes take on the role of translating the viral RNA or DNA into proteins that are essential for the virus. For RNA viruses, the replication process often involves the synthesis of a complementary strand. Picture this as a painter creating a replica of an original masterpiece, using the same colors and techniques, but producing something unique. The host's enzymes, particularly RNA-dependent RNA polymerase, play a vital role in this process, acting as the brush that brings the viral vision to life. Once the viral proteins are synthesized, they are assembled along with the newly replicated genetic material to form new viral particles. This assembly occurs in specific regions of the host cell, often at the endoplasmic reticulum or Golgi apparatus. The viral components come together like pieces of a puzzle, guided by the intricate instructions encoded within the viral genome. As these new viral particles are formed, they prepare for the final step of the replication cycle: release. Release can occur through two primary mechanisms: lysis and budding. In the case of lysis, the host cell is directly destroyed, much like a balloon bursting under pressure, releasing a flood of new viral particles into the surrounding environment. This method, while effective for the virus, can have dire consequences for the host cell, leading to cell death and triggering inflammatory responses. Alternatively, budding allows the virus to exit the host cell more subtly, enveloping itself in a portion of the host membrane. This process is akin to slipping out the back door, leaving the host cell intact while still ensuring the virus's escape. To fully grasp the magnitude of viral replication, one must consider how viruses hijack host cellular processes. Viruses exploit the host's metabolic pathways, commandeering resources that would typically be reserved for the cell's own functions. This manipulation can disrupt vital cellular processes, leading to a range of consequences for the host. For instance, the synthesis of viral proteins requires an abundance of amino acids, which the virus extracts from the host's reserves. As the virus continues to replicate, the host cell finds itself starved of essential resources, creating a scenario where the host's health is compromised. Enzymes play a critical role throughout the viral replication cycle. These biological catalysts are responsible for various reactions that facilitate the synthesis of viral components. For example, reverse transcriptase is an enzyme crucial for retroviruses, converting viral RNA into DNA. This reverse transcription is a pivotal moment, as it allows the viral genetic material to integrate into the host's genome, ensuring that future generations of cells carry the viral instructions. Imagine this enzyme as a skilled translator, converting messages from one language to another, enabling communication between the virus and the host. As the virus takes over the host machinery, it can lead to a myriad of consequences for the health of the infected cell. The rapid production of viral particles can overwhelm the cellular systems, leading to apoptosis or programmed cell death, in an effort to eliminate the intracellular threat. This process, however, is not always straightforward. Some viruses have evolved mechanisms to evade the host's immune response, prolonging the life of the infected cell while continuing to replicate. In these cases, the host may experience chronic infections, where the virus remains hidden, silently replicating and waiting for the opportune moment to strike again. Consider the quote from a renowned scientist: "Viruses are not simply the enemy, they are part of our world, influencing the very fabric of life." This perspective invites us to reflect on the complex relationship between viruses and their hosts. While they can wreak havoc on cellular health, they also play significant roles in ecosystems and evolutionary processes. Viruses contribute to genetic diversity. acting as agents of change that can drive evolution in both microbial and multicellular life. The consequences of viral replication extend beyond individual cells. They can influence entire populations and ecosystems. When viruses infect a host, they can alter the balance of microbial communities, impacting nutrient cycling and energy flow. Imagine a single viral infection cascading through an ecosystem, reshaping the interactions among various organisms, much like the ripples created by a stone thrown into a tranquil pond. As we explore the world of viral replication, we begin to appreciate the exquisite balance between life and the forces that threaten it. Each step in the viral replication cycle unveils a story of survival, adaptation, and resilience. The intricate dance of viral particles, host cells, and enzymes creates a dynamic interplay that shapes the biological landscape. Within this landscape, one wonders, how does the host respond to such relentless invaders? The immune system, a remarkable defense mechanism, springs into action, recognizing viral components and mounting a counteroffensive. This battle between virus and host is ongoing, a testament to the ever-evolving nature of life itself. The intricate strategies employed by both sides highlight the complexity of existence, where survival often hinges on adaptability and resilience. As we contemplate the mechanisms of viral replication, we are reminded of the delicate balance that sustains life. Each virus, in its quest for survival, also contributes to the broader narrative of biological interaction. The relationship between host and virus is a testament to the complexity of life, where even the smallest entities can wield significant influence over living systems. In the grand tapestry of life, viruses occupy a unique niche, serving as both destroyers and creators. They challenge our understanding of health and disease, prompting us to reconsider our place within the web of life. As we continue to examine the intricacies of viral replication, we uncover not only the vulnerabilities of our cells, but also the resilience of life itself. Each interaction, each replication cycle, tells a story of survival, adaptation, and the ongoing quest for existence. The intricate process of viral replication does not merely halt at the assembly of new viral particles. It weaves a deeper narrative involving the consequences of these actions on the host cell's health. As viral particles coalesce and prepare for release, the host cell often finds itself in a precarious situation, caught in a cycle of depletion and disarray. When we consider the effects of viral replication on cell health, it's essential to recognize that viruses are not just passive invaders. They are active agents that influence the host's metabolic pathways. The energy that the host cell typically allocates for its growth, repair, and everyday functions is rerouted to support viral replication. This diversion can lead to a state of cellular distress, as the host cell struggles to maintain its own vitality amid the viral onslaught. Imagine a small factory suddenly compelled to produce an overwhelming quantity of a product for an external entity, neglecting its core operations. For instance, the adenovirus is known to induce the synthesis of specific proteins that disrupt the normal cell cycle. By interfering with the host's regulatory mechanisms, the virus can manipulate the cell into a state of continuous division, facilitating its own replication while jeopardizing the host's typical processes. This can lead to the development of tumors, as the balance between cell proliferation and death is thrown into disarray. Furthermore, the relentless pursuit of replication can trigger inflammatory responses within the host. The immune system, perceiving the viral presence as a threat, mobilizes to combat the invasion. This response, while protective in nature, can inadvertently cause further damage to the host. The inflammation that ensues may lead to tissue damage, contributing to symptoms associated with viral infections, such as fever and fatigue. Reflecting on this interplay, one might ask, how does the host's immune system discern between friend and foe? The answer lies in the sophisticated recognition systems developed over millennia. Host cells are equipped with receptors that detect viral components, activating immune pathways that initiate a response. This recognition is akin to a security system identifying an intruder, setting off alarms and dispatching responders to neutralize the threat. Moreover, some viruses have evolved mechanisms to evade the immune response. The human immunodeficiency virus, for instance, employs tactics to hide from immune surveillance, allowing it to persist within the host for extended periods. This stealthy replication can lead to chronic infections, where the virus remains dormant, yet capable of re-emerging and wreaking havoc when the host's defenses are compromised. As we ponder these dynamics, it is fascinating to consider the implications of viral replication not only on individual cells, but also on the broader ecosystem. Viruses can influence microbial diversity, shaping communities in the soil, oceans, and even within our own bodies. This influence can alter nutrient cycles and energy flow, demonstrating the interconnectedness of life forms. In this way, viruses can serve as agents of change, driving evolution and adaptation in ways we are only beginning to understand. A renowned virologist once remarked, "Viruses are the ultimate survivors." This perspective invites us to view viruses not only as adversaries, but also as integral components of biological ecosystems. Their replication and interaction with hosts can lead to unforeseen consequences that echo through the fabric of life, shaping both microbial and multicellular organisms alike. Imagining this intricate web, one can visualize the dance of life and death, creation and destruction, all orchestrated by these minuscule entities. Each viral replication cycle unfolds a narrative filled with resilience and adaptability, illustrating how even the smallest actors can wield immense power. In contemplating the journey of a virus, we are reminded of the duality of existence. While viral replication can lead to cell death and disease, it also serves as a catalyst for evolutionary change. This complex relationship prompts us to reflect on the balance of life, where each interaction, no matter how small, contributes to the greater narrative of survival. As we delve into the consequences of viral replication for host cell health, we uncover the layers of complexity that define life itself. Each step of the viral life cycle reveals not just a story of infection, but also a tale of adaptation, resilience, and the relentless drive to exist. The interplay between virus and host is a testament to the enduring struggle for survival, where even the most formidable challenges can lead to transformative outcomes. In this grand narrative, it becomes increasingly clear that the relationship between viruses and their hosts is not merely one of conflict, but one of coexistence and evolution. As we continue to explore the myriad ways in which viral replication shapes health and ecosystems, we unveil a deeper understanding of life's intricate processes, where every thread contributes to the rich tapestry of existence. Chapter 7: The Immune Response, Host Defenses Against Viruses. As we explore the intricate world of the immune system, we begin to uncover the remarkable defenses it employs against viral infections. This complex network, comprised of various cells and proteins, functions tirelessly to protect the body from invaders. When a virus breaches the outer defenses, it triggers an immediate response, a cascade of events that unfold with precision. Imagine the immune system as a well-rehearsed symphony, each component playing its role in harmony to counteract the viral assault. The first step in this defensive response is the recognition of viral components by immune cells. Specialized cells, known as antigen-presenting cells, play a crucial role in this recognition process. These cells act as vigilant sentinels, continuously surveying their environment for signs of infection. When they encounter a virus, they ingest it and break it down into smaller fragments, known as antigens. These antigens are then presented on the surface of the antigen-presenting cells, akin to a flag signaling an alert. This presentation is essential for alerting other immune cells, particularly T cells, to the presence of the invader. Imagine the moment a T cell encounters the presented viral antigen. It is as if the T cell receives a personal invitation to the battlefield, signaling that it must spring into action. There are two primary types of T cells involved in this response: helper T cells and cytotoxic T cells. Helper T cells, once activated, release signaling molecules that recruit and activate other immune cells, amplifying the response. Cytotoxic T cells, on the other hand, are the assassins of the immune system. They seek out and destroy infected cells, recognizing them through the specific antigens displayed on their surfaces. The interaction between T cells and the virus is fascinating. When a cytotoxic T cell binds to an infected cell, it releases perforins, which create pores in the target cell's membrane. This action allows granzymes, enzymes that induce apoptosis or programmed cell death, to enter the infected cell. Picture this as a precision strike, targeting only those cells that harbor the invader, ensuring that healthy cells remain unscathed. In tandem with T cells, antibodies produced by B cells play a pivotal role in the immune response. When B cells encounter free-floating viruses or viral particles, they undergo a transformation, becoming plasma cells that produce antibodies specific to the viral antigens. These antibodies act like sentinels, binding to the virus and neutralizing its ability to infect new cells. Imagine them as protective shields, blocking the viral entry points and marking the viruses for destruction by other immune cells. The mechanisms of action employed by antibodies are diverse. They can neutralize viruses, agglutinate viral particles to prevent their spread, or activate the complement system, a group of proteins that enhances the ability of antibodies to clear pathogens. This multifaceted approach illustrates the adaptive nature of the immune response, allowing it to tackle a wide array of viral threats. However, the relationship between viruses and the immune system is not one of simple warfare. Viruses have evolved a remarkable array of strategies to evade detection and thwart the immune response. Some viruses, like the influenza virus, can change their surface proteins rapidly, a phenomenon known as antigenic drift. This ability to alter their appearance makes it difficult for the immune system to recognize and target them effectively. Consider the human immunodeficiency virus, or HIV. This virus employs a more insidious strategy, attacking the very cells that are meant to protect the body. By targeting and destroying helper T cells, HIV undermines the immune system's defenses, rendering the body vulnerable to opportunistic infections. This stealthy approach highlights the ongoing arms race between viruses and the immune system, as each adapts and evolves in response to the other. Furthermore, some viruses can enter a latent phase, where they remain dormant within host cells, evading immune detection. The herpes simplex virus is a prime example, lying inactive in the nervous system until triggered by stress or other factors to reemerge. This ability to hide poses significant challenges for the immune system, which must remain vigilant even in the absence of overt signs of infection. The immune response is a testament to the resilience and adaptability of life itself. It is a dynamic interplay of recognition, action, and counteraction. Yet, even with its incredible capabilities, there are times when the immune system falters, and viruses succeed in establishing infections. This duality raises intriguing questions about the balance of power in this microscopic world. What makes some individuals more susceptible to viral infections than others? How do genetics and environmental factors influence the effectiveness of the immune response? As we contemplate these questions, we begin to understand the complexity of the immune response. It is not merely a series of steps, but a finely tuned orchestration of cellular interactions and signaling pathways, all working toward a common goal: the preservation of health. Each encounter with a virus serves as a lesson, shaping the immune system's memory and enhancing its ability to respond more effectively in the future. This adaptive immunity is the cornerstone of vaccinations, a remarkable achievement in public health. Vaccines expose the immune system to harmless components of a virus, training it to recognize and combat the real threat should it arise. The concept of immunological memory illustrates the profound intelligence of the immune system, allowing it to remember past encounters and respond with greater speed and efficiency. As we delve into the nuances of the immune response, we find ourselves marveling at the intricate web of interactions that sustain life. The immune system, with its myriad components and functions, is a reflection of the delicate balance that governs our existence. Each cell, each signaling molecule, contributes to a grand design that has evolved over millennia, ensuring that we are equipped to face the challenges posed by the viral world. In this continuous battle, the immune system emerges not merely as a defender, but as a testament to the resilience of life itself. It invites us to appreciate the wonders of biology, to contemplate the intricate systems that sustain us, and to recognize the delicate interplay between vulnerability and strength. Each viral encounter shapes our understanding of what it means to be alive, fostering an appreciation for the extraordinary complexities that lie within us. The immune response is not merely a static defense. It is a dynamic and adaptable system capable of learning from past encounters. This remarkable ability is primarily attributed to memory cells, which arise from the initial immune reaction. After a successful battle against a virus, a subset of the activated B and T cells persist long after the infection has cleared. These memory cells stand ready, like sentinels, to spring into action upon future exposure to the same virus. Imagine these memory cells as a library of experiences, each containing detailed accounts of previous infections. Upon re-encountering their viral nemesis, they can mount a rapid and robust response, often neutralizing the threat before it establishes a foothold. The role of memory cells highlights the profound connection between the immune system and our health. For instance, vaccines leverage this innate ability of the immune system to create lasting immunity. Through the introduction of harmless viral components, vaccines prepare the immune system for potential future assaults. This training process is akin to rehearsing a play. When the curtain rises for the real performance, the actors are ready to deliver their best. However, as effective as the immune system may be, it is not infallible. The intricate dance of immune responses can sometimes lead to missteps, resulting in autoimmune diseases, where the body mistakenly attacks its own cells. This paradox raises a thought-provoking question. How does the immune system distinguish between friend and foe? The answer lies in the complex recognition mechanisms that involve a multitude of signals and checkpoints designed to ensure accuracy. Yet in some cases, these safeguards fail, leading to unintended consequences. Consider the autoimmune condition known as lupus, where the immune system erroneously targets the body's tissues. This misjudgment can cause widespread inflammation and damage, illustrating the delicate balance that must be maintained. In essence, the immune system must strike a harmony between vigilance and tolerance, a task that is both vital and challenging. In addition to the challenges posed by the immune system's own complexities, viruses employ an array of cunning strategies to evade immune defenses. Some viruses possess the ability to mimic host molecules, effectively masquerading as harmless entities. This clever disguise can confuse the immune system, allowing the virus to persist within the host. Imagine a thief wearing a disguise, seamlessly blending into a crowd, making detection nearly impossible. Moreover, certain viruses can interfere with the signaling pathways that activate immune responses. For example, the hepatitis C virus has been shown to disrupt the signaling of interferons, proteins that play a pivotal role in orchestrating the immune response to viral infections. Such interference not only weakens the immune reaction, but can also lead to chronic infections, as the virus remains hidden from the vigilant eyes of the immune system. The ongoing battle between viruses and the immune system is a testament to the evolutionary arms race that shapes all life. Viruses, in their relentless pursuit of survival, continuously adapt and evolve, developing new mechanisms to avoid detection and destruction. For instance, the influenza virus is notorious for its ability to undergo rapid genetic changes, resulting in new strains that can evade the immune memory established by previous infections or vaccinations. This phenomenon is why flu vaccines are often updated annually, ensuring that they remain effective against the most current strains circulating in the population. As we contemplate the complexities of this interplay, we are drawn to the resilience of the immune system. Despite the myriad challenges it faces, it continues to evolve and adapt, learning from each encounter. This adaptability is not merely a biological curiosity, it has profound implications for public health and disease prevention. Understanding these mechanisms allows researchers to develop better vaccines and therapeutic strategies to bolster our defenses against viral threats. In the midst of this ongoing struggle, the stories of scientists who have dedicated their lives to unraveling the mysteries of the immune response emerge as a source of inspiration. Figures like Louis Pasteur, who pioneered the field of immunology, demonstrated the power of vaccines to prevent devastating diseases. His work laid the foundation for our modern understanding of how the immune system can be trained to recognize and combat pathogens. Pasteur once said that science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. This belief in the unifying power of scientific discovery continues to drive researchers today. As we explore the depths of immune responses, we begin to appreciate the intricate web of interactions that define our fight against viruses. Each encounter is not just a battle, but a complex negotiation between the immune system and its adversaries. The lessons learned from these encounters shape not only individual health outcomes, but also the collective resilience of populations in the face of emerging viral threats. In this ongoing saga of survival, it becomes clear that the immune system is not just a collection of cells and molecules working in isolation. Rather, it is a remarkable system built on cooperation and communication, where each component plays a vital role in ensuring the integrity of life itself. This harmony invites us to reflect on our own interconnectedness, reminding us that the health of one is, in many ways, the health of all. As we continue to unravel the complexities of viral interactions with the immune system, we must remain vigilant and curious. The journey of discovery is far from over, and with each new revelation, we inch closer to understanding the delicate balance that governs our existence in a world teeming with unseen adversaries. What new strategies will researchers uncover? How will our understanding of the immune response shape the future of medicine? As we ponder these questions, we are reminded of the profound resilience inherent in life itself, a resilience that we must safeguard and nurture as we navigate the challenges that lie ahead. Chapter 8: Viral Spread Within the Host. As we delve into the mechanisms by which viruses spread within a host, we uncover a remarkable journey marked by complexity and precision. Once a virus has successfully invaded a host cell, its ability to disseminate throughout the body becomes paramount for its survival and propagation. This viral spread is not a random process. Rather, it is orchestrated through a series of well-defined mechanisms that allow the virus to exploit the host's biological systems. One of the primary routes of viral transmission within the host is through the bloodstream. Picture the circulatory system as a vast network of highways, with red blood cells serving as delivery vehicles. When a virus enters the bloodstream, it can ride along this network, reaching distant tissues and organs. This method is particularly effective for viruses such as the human immunodeficiency virus, which can circulate in the blood and infect immune cells, establishing a foothold in the body. The ability of viruses to utilize the bloodstream highlights their adaptability and the intricate relationship they forge with their hosts. Another route of transmission is through direct cellular contact. Viruses can spread from one infected cell to adjacent uninfected cells through a process known as cell-to-cell spread. Imagine this as a whispering chain, where the viral particles pass directly from one cell to another, evading some of the immune defenses that would typically target free-floating viruses in the bloodstream. This method can be particularly efficient, as it allows the virus to maintain a localized infection while simultaneously spreading without attracting immediate attention from the immune system. Understanding the nuances of viral spread is critical, not only for appreciating the biology of viruses, but also for developing effective treatment strategies. The amount of virus present in the body, often referred to as the viral load, plays a pivotal role in disease progression. High viral loads can overwhelm the immune response, leading to more severe disease outcomes. For instance, individuals with a high viral load of the influenza virus often experience more intense symptoms and longer illness durations. This correlation raises important questions. How does the viral load affect the body's defenses? Can we manipulate this load to improve treatment outcomes? The immune system's response is intricately linked to the viral load. When faced with a high concentration of viral particles, the immune system must mount a more vigorous attack, often leading to inflammation and tissue damage. This phenomenon can be likened to a fire brigade responding to a raging inferno, deploying all available resources to extinguish the flames. Yet, this aggressive response can sometimes be counterproductive, resulting in collateral damage to healthy tissues. Therefore, understanding the interplay between viral load and immune response is vital for crafting therapeutic interventions. Moreover, the specifics of viral dissemination can vary significantly among different viruses. For example, respiratory viruses, such as the one causing COVID-19, spread through aerosolized droplets, allowing them to infect new hosts through inhalation. Once inside the respiratory tract, these viruses can quickly replicate and spread to neighboring cells. The rapidity of this process can lead to a swift increase in viral load, which in turn influences the severity of respiratory symptoms. The ability to spread efficiently through the air exemplifies how certain viruses have adapted to exploit the environment of their hosts for maximum impact. In contrast, some viruses prefer to spread through more localized routes, using direct cell-to-cell transfer to minimize detection by the immune system. Herpes simplex virus, for instance, can remain dormant in nerve cells, reactivating later to spread along nerve pathways. This ability to hide and then reemerge illustrates the dynamic relationship between a virus and its host, marked by periods of dormancy followed by active replication and spread. As we consider the implications of viral spread, the significance of understanding these mechanisms becomes increasingly clear. Knowledge of how viruses disseminate within the host informs treatment strategies, guiding the development of antiviral therapies aimed at disrupting the viral life cycle. By targeting specific stages of viral replication or spread, researchers can devise interventions that not only reduce viral load, but also mitigate the symptoms associated with viral infections. Moreover, understanding the routes of transmission within the host allows healthcare professionals to better predict disease progression and tailor patient care accordingly. For instance, if a virus is known to spread rapidly through the bloodstream, early intervention may be critical in preventing severe outcomes. This insight can empower clinicians to implement timely therapeutic measures, enhancing patient outcomes and potentially saving lives. The ongoing research into viral spread and its implications for treatment continues to reveal the sophistication of these microscopic invaders and their interactions with the host. Each discovery adds a new layer to our understanding, inviting us to imagine the unseen battles waged within our bodies. As we continue to explore the intricacies of viral life, we must remain aware of the delicate balance that exists between host and pathogen, a dance defined by survival, adaptation, and the relentless pursuit of existence. In contemplating the viral spread within a host, we are reminded of the profound interconnectedness of life, where even the tiniest entities can wield immense power over biological systems. The tapestry of life is woven with threads of competition and cooperation, resilience and vulnerability. As we navigate the complexities of viral dissemination, we are not just learning about viruses, we are gaining insights into the very essence of life itself. As we explore the intricate pathways of viral dissemination, we come to understand the various routes through which viruses can traverse the host's body. Beyond the bloodstream and direct cellular contact, lymphatic pathways emerge as another critical avenue for viral spread. The lymphatic system, comprising a network of vessels and nodes, acts as a conduit for immune cells and interstitial fluid. This system provides a unique environment for viruses to exploit. Picture the lymphatic vessels as winding rivers, carrying along not only immune cells, but also viral particles that can infiltrate lymph nodes, where they may proliferate and potentially evade immune detection. Viruses such as Epstein-Barr virus are known to utilize the lymphatic system effectively. Once in the lymph nodes, they can infect B cells, a type of immune cell responsible for producing antibodies. This interaction exemplifies the dual nature of viruses, which can manipulate the host's immune response to their advantage. By establishing an infection in immune cells, the virus may not only persist, but also spread to other tissues, complicating the host's ability to mount an effective defense. Imagine a skilled infiltrator, operating within the ranks of the defenders, sowing the seeds of its own survival. Moreover, the respiratory tract serves as another vital route for viral transmission within the host. Respiratory viruses, including those responsible for influenza and COVID-19, thrive in the epithelial cells lining the airways. Once inhaled, these viruses can quickly replicate and spread throughout the respiratory system. The nature of the respiratory epithelium, with its rich supply of mucus and cilia, provides both a challenge and an opportunity for viral spread. The cilia, tiny hair-like structures that help clear pathogens, can be outmaneuvered by viruses that have adapted to evade this defense. Imagine this scenario as a game of cat and mouse, where the virus utilizes clever strategies to bypass the host's protective mechanisms. The impact of viral load becomes increasingly significant as we consider the various routes of spread. Each viral particle represents a potential threat, and as the viral load increases, so too does the likelihood of severe disease outcomes. High viral loads can lead to a greater number of infected cells, overwhelming the immune response and resulting in increased tissue damage. This relationship can be likened to a dam under pressure. As the flow of water increases, the structure becomes more susceptible to failure. The body's defenses, while robust, have limits. Understanding these thresholds is crucial for anticipating disease progression. As we reflect on the implications of viral load, we find ourselves asking, how can we harness this knowledge to inform treatment strategies? The answer lies in a nuanced understanding of the viral life cycle and the dynamics of host responses. For example, antiviral treatments that reduce viral replication can subsequently lower viral load, thereby alleviating symptoms and improving outcomes. The goal becomes clear: to disrupt the viral life cycle at strategic points, limiting its ability to spread and cause harm. The relationship between viral spread and treatment strategies extends beyond merely reducing viral load. The timing of intervention is vital. Early detection and treatment of viral infections can significantly alter the course of disease. Consider how swift action can make all the difference. Much like a gardener who acts quickly to prune a budding weed before it takes hold, timely therapeutic measures can prevent the escalation of infection. This proactive approach underscores the importance of understanding the mechanisms of viral dissemination, allowing for targeted interventions that mitigate risk. As researchers delve deeper into these mechanisms, they uncover the complex interplay between viral spread and host responses. Insights gained from studying renowned viruses can illuminate potential pathways for intervention. For instance, the lessons learned from the Ebola virus during past outbreaks have shaped our understanding of how to contain viral spread within the host. By understanding its transmission routes and the dynamics of viral load, scientists developed vaccines and treatments that have proven effective in mitigating future outbreaks. The pursuit of knowledge in this realm is relentless. Each new discovery brings us closer to unraveling the mysteries of viral behavior and its effects on human health. The journey is akin to charting uncharted waters, where each wave of understanding leads to new horizons of possibility. As we continue to explore the nuances of viral spread, we are reminded of the importance of collaboration across disciplines. Scientists, clinicians, and public health experts must work in concert, leveraging their unique perspectives to develop comprehensive strategies that address viral infections. In this ongoing quest, we must also consider the role of public awareness and education. An informed populace can better understand the importance of preventative measures, such as vaccinations and hygiene practices, that limit the spread of viruses. Imagine communities coming together, united in their efforts to protect one another, creating a collective shield against potential outbreaks. The power of knowledge and awareness can transform societal responses to viral threats, fostering resilience in the face of adversity. As we navigate the complexities of viral dissemination, it becomes increasingly evident that each virus possesses its own unique strategies and adaptations. While some viruses thrive in the bloodstream, others may prefer the lymphatic system or respiratory pathways. This diversity is a testament to the evolutionary pressures that shape viral behavior, prompting us to ask, What drives this adaptability? The answer lies in the relentless pursuit of survival, where viruses continuously adapt to their host environments, striving for dominance. Understanding these mechanisms extends beyond academic curiosity. It holds profound implications for the future of medicine. By unraveling the intricacies of viral spread, we pave the way for innovative treatment approaches that can outsmart these elusive adversaries. The road ahead is filled with challenges, but with each insight gained, we move closer to a future where viral infections can be effectively managed and controlled. The delicate balance between host and pathogen remains a central theme, reminding us of the intricate web of life that connects us all. Chapter 9: The Impact of Viruses on Host Cells and Tissues. As we turn our attention to the intricate relationship between viruses and their host cells, we begin to understand how these microscopic entities influence cellular function. When a virus infects a host, it does not merely invade. It orchestrates a profound transformation within the host cell, commandeering its resources for its own replication. Imagine a delicate balance being disrupted, where the normal rhythm of cellular activity is overshadowed by the viral presence. Infected cells can exhibit a plethora of changes in their behavior and structure. Viral infections can alter cellular metabolism, redirecting energy and resources to support viral replication rather than the cell's usual functions. This hijacking can lead to cellular stress and dysfunction, as the virus manipulates the host's machinery to produce viral components. Some cells may even begin to produce viral proteins instead of their own, marking a dramatic change in their identity. What happens to the host cells as they become embroiled in this viral takeover? The consequences can be dire. When the cellular functions are overwhelmed, it can lead to cell death, a phenomenon that plays a critical role in viral pathogenesis. For instance, some viruses induce apoptosis, a programmed cell death process that serves as a defense mechanism against infection. This is like a soldier sacrificing themselves for the greater good, ensuring that the virus does not have the opportunity to spread further. Yet, not all viral infections lead to immediate cell death. Some viruses have evolved strategies to evade detection and prolong the life of their host cells, allowing for continued viral replication. Imagine a thief who has managed to convince the homeowner to let them stay indefinitely, using charm and manipulation to avoid being kicked out. This strategy can result in chronic infections, where the virus remains in the host for extended periods, subtly altering the host's cellular environment. The long-term effects of viral infections on host tissues can be profound. Even after the initial infection is cleared, remnants of viral activity can persist, leaving lasting damage. For example, the hepatitis B virus can lead to chronic liver disease, with inflammation and scarring that may progress to cirrhosis or liver cancer. Similarly, the human papillomavirus can cause cellular changes that lead to cancers of the cervix and other tissues long after the initial infection has resolved. The lingering effects of these infections prompt us to ask, how do viruses manipulate host cellular pathways to achieve such long-lasting consequences? What is fascinating is how these viral infections can alter the very fabric of host tissues. Take, for instance, the impact of the Epstein-Barr virus, which is linked to several diseases, including mononucleosis and certain types of cancer. This virus can lead to the proliferation of infected B cells, disrupting normal immune function and potentially leading to malignancies. The virus's ability to alter cellular signaling pathways not only allows it to thrive, but also transforms the host's tissue architecture. Some of the most notorious viral infections showcase these impacts vividly. Consider the influenza virus, which can cause severe respiratory illness. The virus replicates extensively in the epithelial cells of the respiratory tract, leading to cell death and the characteristic symptoms of the flu. The body's immune response to this cell death can further exacerbate tissue damage, highlighting the intricate interplay between virus and host. Another striking example is the human immunodeficiency virus, or HIV. This virus primarily targets CD4 T cells, crucial components of the immune system. By depleting these cells, HIV undermines the host's ability to mount an effective immune response, leading to increased susceptibility to opportunistic infections and malignancies. The long-term battle between HIV and the immune system is a vivid illustration of how a virus can shape the fate of host tissues over time. The impact of viral infections extends beyond the individual cells they invade. The consequences ripple through the host's entire system, leading to systemic effects. For example, the Zika virus, while often presenting mild symptoms, can lead to significant neurological consequences, especially in fetuses. The virus can disrupt normal brain development, leading to microcephaly and other severe birth defects. This highlights a profound and tragic outcome of viral persistence and its ability to profoundly affect tissue development. The stories of these viruses remind us of the delicate balance within our bodies and the intricate dance between host and pathogen. As we reflect on the myriad ways viruses can influence cellular function, we are reminded of the resilience of life and the ongoing struggle between viral invaders and the host's defenses. Each encounter is a story of survival, adaptation, and, at times, tragic consequences. Ultimately, the impact of viruses on host cells and tissues is a testament to the complexity of life itself. As we continue to explore this dynamic interplay, we uncover not only the mechanisms by which viruses operate, but also the profound effects they can have on the health and longevity of their hosts. What does the future hold as we deepen our understanding of these interactions? Only time will reveal the answers, but one thing is certain. The dance between viruses and their hosts will persist, weaving a narrative of challenge and resilience that continues to unfold in the biological theater of life. As we explore further into the impact of viral infections on host cells and tissues, we encounter a profound aspect of viral pathogenesis, the role of cell death. This phenomenon is not merely a byproduct of infection. It is intricately woven into the virus's strategy to ensure its survival and proliferation. When a virus invades a host cell, it often triggers a cascade leading to cell death, whether through apoptosis or necrosis. Apoptosis, the programmed cell death, serves as an essential defense mechanism. It is an elegant process, akin to a well-coordinated evacuation, where the infected cell dismantles itself to prevent the virus from spreading to neighboring cells. Imagine a building on fire, where the inhabitants recognize the danger and choose to leave before the flames engulf them. This is the essence of apoptosis, a self-sacrificial act aimed at safeguarding the broader community. Yet, in some instances, viruses can manipulate this process to their advantage. Certain viral proteins can inhibit apoptosis, allowing the infected cell to persist and, in turn, continue producing viral particles. This manipulation can fuel chronic infections, leading to long-term consequences for the host. The dynamics of cell death extend beyond individual cells. When a significant number of cells die, it can create a cascade of effects on the surrounding tissue, leading to inflammation and further cellular stress. This inflammatory response is the body's attempt to combat the viral invader, but it can also exacerbate tissue damage. For example, in cases of severe viral infections like those caused by the dengue virus, the immune response can lead to a condition known as dengue hemorrhagic fever. characterized by increased vascular permeability and bleeding. The consequences of such viral pathogenesis illustrate the delicate interplay between host defenses and viral strategies. Moving on to the long-term effects of viral infections, we find that these viruses can leave indelible marks on host tissues even after the acute phase of infection has resolved. The insidious nature of certain viruses allows them to persist in a latent state, subtly influencing the host's cellular environment and health. Take the herpes simplex virus as an example. After the initial infection, this virus can establish latency in sensory neurons, remaining dormant until reactivated by factors such as stress or immunosuppression. Each reactivation can lead to lesions and discomfort, illustrating how the virus maintains a foothold in the host's biology. The long-term consequences extend to chronic inflammation and immune dysregulation, which can ultimately result in tissue damage. The hepatitis C virus is another prime example, where persistent infection can lead to chronic liver disease. Even after the virus is cleared, the scars left on the liver can predispose individuals to cirrhosis or even hepatocellular carcinoma. This raises an essential question. How do these viruses manipulate host cellular pathways to drive such lasting effects? Consider the interplay between the immune system and viral infections. The immune response, while crucial for controlling the infection, can also lead to collateral damage. In the case of chronic viral infections, the immune system may become exhausted, leading to a state of chronic inflammation. This, in turn, can create a permissive environment for the development of malignancies. The link between viral infections and cancer is particularly striking, leading researchers to investigate the mechanisms through which viruses can induce cellular transformation. The human papillomavirus serves as a poignant example in this context. Certain strains of this virus are linked to the development of cervical and other cancers. The viral proteins E6 and E7 can interfere with the host's cell cycle regulation, promoting uncontrolled cellular proliferation. Imagine a garden where weeds are allowed to flourish unchecked. This analogy captures how the virus can disrupt the normal balance of cellular growth and division, leading to malignancy. Moreover, we cannot overlook the impact of viral infections on the neurological system. The rabies virus, for instance, travels along neural pathways to the brain, leading to severe neurological symptoms and ultimately death if untreated. The manner in which this virus influences neuronal function underscores the profound effects viruses can have beyond mere tissue damage. They can alter behavior, cognition, and bodily control. The long-lasting impact of viral infections on host tissues also encompasses the respiratory system. The SARS-CoV-2 virus, responsible for the COVID-19 pandemic, has demonstrated that even after the acute phase of infection, many individuals experience lingering symptoms, a condition often referred to as long COVID. This phenomenon includes a range of symptoms such as fatigue, respiratory difficulties, and cognitive impairments. The underlying mechanisms remain an area of active research, but it highlights the capacity of viruses to inflict enduring damage on host tissues. In summary, the exploration of viral infections reveals a complex narrative of interaction between viruses and their hosts. The ability of viruses to induce cell death, manipulate immune responses, and create lasting impact on tissues illustrates the intricate balance of life and disease. As we continue to unravel these connections, we gain insight not just into the viruses themselves, but into the resilience and adaptability of the host organism. Each discovery brings us closer to understanding how to mitigate the effects of these viral invaders, paving the way for future advancements in medicine and public health. The impact of viral infections on host cells and tissues is a multifaceted narrative, revealing both immediate and long-term consequences that shape the health of the host. As we consider the intricate mechanisms at play, we find that cell death is not merely an aftermath of infection, but rather a crucial element of viral pathogenesis. For many viruses, inducing cell death serves as a strategy to both propagate their existence and manipulate the host's immune response. When we think about the role of cell death, it invites us to imagine a battlefield where infected cells are akin to soldiers facing a relentless enemy. In this context, apoptosis acts as a noble sacrifice, a controlled retreat to prevent the virus from spreading. Yet, this is not the only narrative. Some viruses, such as the hepatitis C virus, employ cunning tactics to evade the immune system, allowing them to persist in the host for years. This capacity to manipulate cellular pathways for self-preservation is a testament to the virus's evolutionary prowess. The long-term consequences of viral infections extend into the very fabric of host tissues. Even after acute infections have resolved, the remnants of viral activity can linger, inciting chronic inflammation and tissue remodeling. This is particularly evident with the human immunodeficiency virus, which establishes a persistent infection by integrating its genetic material into the host genome. Such integration can lead to ongoing immune evasion and contributes to the long-term health challenges faced by those living with HIV. Consider also the case of the Epstein-Barr virus, notorious for its role in infectious mononucleosis and its association with various malignancies. The virus's ability to induce cellular proliferation and alter immune regulation showcases how viral infections can lead to significant and lasting changes in host tissues. The interplay between viral persistence and cellular transformation raises important questions about the mechanisms by which these viruses manipulate host signaling pathways. The impact of viruses is not confined to the surface of tissues. It penetrates deeply into the host's biological systems, influencing everything from metabolic processes to immune responses. Take the example of the respiratory syncytial virus, which primarily affects the lungs. This virus leads to acute respiratory infections, but the inflammation it incites can result in long-lasting changes to lung tissue, contributing to conditions like asthma in susceptible individuals. The transient nature of the infection belies its potential for long-term consequences, prompting us to reflect on how even brief encounters with viruses can leave indelible marks on our health. Furthermore, we see the profound implications of viral infections on the nervous system. The rabies virus, with its notorious ability to traverse neural pathways, highlights the vulnerability of the nervous system to viral invasion. Once the virus enters the brain, it can lead to severe neurological deficits and ultimately death. This journey through the nervous system underscores the extraordinary adaptability of viruses as they exploit the host's own networks for their propagation. In considering these cases, we must also acknowledge the emerging phenomenon of post-viral syndromes. The COVID-19 pandemic has brought renewed attention to the long-term effects of viral infections, with many individuals experiencing lingering symptoms known as long COVID. The relationship between viral infections and chronic health conditions invites us to explore the mechanisms that underpin these enduring effects, which may include immune dysregulation and persistent viral reservoirs. The stories of these viral infections remind us of the delicate balance within our bodies and the intricate interplay between host and pathogen. Each encounter with a virus carries the potential for both immediate and far-reaching consequences, highlighting the need for continued research and understanding. As we navigate this complex landscape, we are reminded of the resilience of life itself and the ongoing struggle to maintain health in the face of such formidable adversaries. In conclusion, the impact of viruses on host cells and tissues represents a rich tapestry of interaction, where the stakes are high for both the invader and the host. The stories of viral infections compel us to consider not only the mechanisms of disease, but also the broader implications for health and well-being. The journey through this chapter reveals the multifaceted ways in which viruses shape the biological landscape, creating a narrative of challenge, adaptation, and survival. As we move forward, the exploration of these themes will undoubtedly yield further insights into the intricate dance between viruses and their hosts, ensuring that the story continues to unfold. Chapter 10: Future Directions: Combating Viral Infections. As we look toward the horizon of scientific discovery, we find ourselves at a pivotal juncture in our battle against viral infections. Current strategies for antiviral therapies and vaccines are a testament to human ingenuity and resilience. With a growing understanding of viral mechanisms, researchers are continually developing innovative ways to combat these elusive foes. Antiviral therapies today can be categorized into several approaches, each targeting different stages of the viral life cycle. Some agents work to inhibit viral entry into host cells, while others disrupt replication or assembly of viral particles. For example, protease inhibitors prevent the virus from processing its proteins, effectively blocking its ability to mature and spread. Imagine these inhibitors as carefully placed roadblocks on the viral highway, preventing it from reaching its destination. Vaccines, on the other hand, have emerged as one of the most powerful tools in our arsenal. They harness the body's immune response to prepare it for future encounters with specific viruses. Vaccines can stimulate both antibody and cellular responses, creating a robust defense system. The success of vaccines against diseases like measles and hepatitis B illustrates the power of preventive medicine. Picture a fortress built not from stone, but from the body's own defenses, ready to repel invaders. Yet, as we celebrate the advancements made, it is crucial to acknowledge the challenges that lie ahead. Developing broad-spectrum antiviral drugs poses a significant hurdle in the fight against viral infections. These drugs, designed to work against a wide range of viruses, must contend with the rapid mutation rates that characterize many viral pathogens. When a virus replicates, small errors can occur in its genetic material, leading to variants that may evade existing treatments. How can we stay ahead of these constantly evolving enemies? Researchers are exploring diverse strategies to tackle this challenge. One promising avenue involves targeting essential viral processes that are common across multiple viruses, such as the viral replication machinery. By focusing on these universal mechanisms, scientists hope to create therapies that can effectively combat a variety of viral infections. Imagine casting a wide net over a turbulent sea, aiming to catch not just one fish but many, regardless of their species. Moreover, the integration of advanced technologies is revolutionizing our approach to viral control. The rise of genomic sequencing has provided unprecedented insights into viral evolution and pathogenesis. By decoding the genetic blueprints of viruses, researchers can identify vulnerabilities that may be exploited for therapeutic purposes. This technological leap is akin to equipping ourselves with powerful binoculars, allowing us to see far beyond the immediate landscape. Artificial intelligence is also making strides in the realm of virology. Machine learning algorithms analyze vast datasets to predict viral outbreaks and evaluate potential antiviral compounds. Imagine a vast library of knowledge, where the wisdom of countless studies converges to illuminate paths forward. These innovations not only accelerate the pace of discovery, but also enhance our capacity to respond to emerging viral threats swiftly. In addition to these advancements, the importance of research into viral pathogenesis cannot be overstated. Understanding how viruses interact with host systems provides critical insights that inform the development of effective treatments and vaccines. Each viral infection unfolds a unique narrative, revealing the complexities of host-virus interactions. For instance, the mechanisms by which viruses evade the immune response are a focal point of research. By studying the tricks that viruses employ, we can devise countermeasures to outsmart them. Consider the work of pioneering scientists who have dedicated their lives to unraveling these mysteries. Their relentless pursuit of knowledge has illuminated pathways previously shrouded in darkness. Each discovery acts as a stepping stone, building a foundation for future breakthroughs. Their contributions remind us that the fight against viral infections is not merely a scientific endeavor, but a collective human effort, drawing on the talents and insights of individuals from diverse backgrounds. Yet, as we venture into this promising future, we must remain vigilant. The challenges we face are formidable, and complacency can be our greatest adversary. The rapid pace of viral evolution serves as a stark reminder that we must continuously refine our strategies. New viruses can emerge from unexpected sources, sometimes crossing species barriers and spreading rapidly. This unpredictability calls for a proactive approach to research and public health. The COVID-19 pandemic has underscored the importance of global collaboration in addressing viral threats. It has shown us that viruses do not respect borders, and our responses must be coordinated and comprehensive. Imagine a world where scientists, healthcare professionals, and policymakers work hand in hand, sharing knowledge and resources to combat viral infections on a global scale. Such collaboration not only enhances our ability to respond to existing threats, but also fortifies our defenses against future outbreaks. In this interconnected landscape, the role of education and public awareness cannot be overlooked. Empowering communities with knowledge about viral infections, prevention strategies, and the importance of vaccination fosters resilience. Imagine a society where individuals are not only informed, but also engaged in the collective effort to safeguard public health. This proactive stance can lead to a more prepared and healthier population. As we continue to navigate this intricate web of challenges and opportunities, the future of antiviral therapies and vaccines shines brightly. With each advancement, we take a step closer to understanding and ultimately controlling viral infections. The marriage of science, technology, and human ingenuity holds the promise of a world where viral threats are met with robust defenses, protecting individuals and communities alike. In this evolving landscape, the synergy between innovation and research lays the groundwork for the next generation of antiviral strategies. The potential for breakthroughs is immense, driven by the passion and determination of those dedicated to this cause. As we embark on this journey, we do so with a sense of hope and purpose, striving to create a healthier future for all. The whispers of possibility echo around us, urging us to push the boundaries of what is known and to embrace the unknown with courage and curiosity. The landscape of antiviral therapies is continually evolving, driven by the pressing need to address emerging viral threats. Current strategies, while effective, must adapt to keep pace with the relentless evolution of viruses. This adaptability is essential, especially as we confront the challenges presented by broad-spectrum antiviral drugs. As we have seen, the rapid mutation rates of many viruses can lead to the emergence of resistant strains, complicating treatment strategies. In our quest for more effective antiviral therapies, we must consider innovative approaches that extend beyond traditional methods. One intriguing strategy involves utilizing host-targeted therapies. Unlike conventional antivirals that directly target the virus, these therapies aim to bolster the host's immune response, enhancing the body's natural defenses against viral infections. Imagine training the body's own army of immune cells to recognize and combat viral invaders with greater efficiency. This approach not only reduces the burden of the virus, but may also minimize the likelihood of resistance developing. Another promising avenue is the exploration of RNA interference, a natural cellular process that can silence specific genes. Researchers are investigating how to harness this mechanism to target viral RNA, effectively shutting down the virus's ability to replicate. Picture this as a precise strike against the virus's genetic blueprint, incapacitating its ability to produce new copies of itself. This targeted approach offers hope for treating infections caused by a variety of viruses, including those that currently lack effective therapies. The importance of research into viral pathogenesis cannot be overstated, especially as we seek to understand the nuances of viral behavior within host systems. Each viral infection tells a unique story, revealing the intricate dynamics between virus and host. By studying these interactions, we can uncover potential vulnerabilities that can be exploited for therapeutic gain. Consider the work of researchers who have studied the molecular interactions between viruses and host cells, shedding light on how certain viruses evade the immune response or manipulate cellular machinery. Take the insights gained from studying the human immunodeficiency virus, which has adapted unique mechanisms to persist within its host. Understanding how HIV integrates into the host genome and evades immune detection has opened new avenues for therapeutic intervention. This knowledge is akin to deciphering a complex code, revealing the underlying principles that govern viral survival and propagation. Yet, the challenges remain daunting. The journey to develop broad-spectrum antiviral drugs is fraught with obstacles, including the need to balance efficacy with safety. As we venture further into this realm, we must remain cognizant of the potential side effects that may arise from targeting both viral and host processes. This delicate balancing act requires ongoing research and vigilance, as we strive to create therapies that are not only effective but also safe for patients. In this rapidly advancing field, technology and innovation play crucial roles in shaping the future of virus control. The integration of cutting-edge technologies is revolutionizing our approach to antiviral drug discovery. High-throughput screening methods allow researchers to rapidly evaluate thousands of compounds for antiviral activity, accelerating the identification of potential therapies. Imagine a bustling laboratory filled with scientists working tirelessly, their efforts fueled by the promise of new discoveries waiting to be uncovered. Furthermore, the advent of structural biology techniques, such as cryo-electron microscopy, has provided unprecedented insights into viral structures at atomic resolution. Understanding the intricate details of viral architecture can inform the design of novel antiviral agents that target specific viral components. These breakthroughs are akin to possessing a detailed map of a complex terrain, guiding researchers as they navigate the uncharted landscape of viral biology. Artificial intelligence also holds immense potential in the realm of virology. Machine learning algorithms can analyze vast data sets, identifying patterns and predicting viral behavior with remarkable accuracy. Picture a vigilant sentinel constantly observing and learning from the data, ready to alert us to potential threats before they materialize. These technological advancements not only enhance our capacity to respond to emerging viral outbreaks, but also facilitate the development of personalized therapies tailored to individual patients. As we explore the future directions of combating viral infections, the importance of interdisciplinary collaboration becomes increasingly evident. The fight against viruses is not confined to the realm of virology alone. It necessitates the integration of knowledge from diverse fields, including immunology, molecular biology, and bioinformatics. Imagine a vibrant ecosystem where experts from various disciplines come together, each contributing their unique insights to tackle the challenges posed by viral infections. This collaborative spirit is exemplified by initiatives such as global research consortia, which unite scientists and healthcare professionals across borders. By sharing data, resources, and expertise, these collaborations amplify our efforts to combat viral threats. The COVID-19 pandemic has highlighted the urgency of such collaborations, as researchers around the world worked in unison to develop vaccines and treatments at an unprecedented pace. Moreover, the role of public health initiatives in disseminating information and promoting vaccination cannot be overstated. Empowering communities with knowledge about viral infections and the importance of preventive measures fosters resilience. Imagine a society where individuals understand the significance of their role in safeguarding public health, actively participating in vaccination campaigns and adopting practices that minimize the spread of viruses. This proactive engagement is crucial as we confront the multifaceted challenges of viral infections. The unpredictability of emerging viruses demands a vigilant and prepared global community. As we look to the future, we must embrace a mindset of adaptability and innovation, continually refining our strategies to stay ahead of viral threats. Through a combination of advanced research, technological innovation, and collaborative efforts, the future of antiviral therapies and vaccines appears more promising than ever. The journey ahead is filled with challenges, but it is also rich with opportunities for discovery and advancement. As we stand on the precipice of this new era in virology, we are reminded of the resilience of the human spirit and our unwavering commitment to safeguarding health and well-being. The path may be complex, but the potential for breakthroughs beckons, urging us onward toward a healthier future for all.